Hello, my name is Bo Zhang. I'm a professor in the Department of Chemistry at the University of Washington. My student and I, we work in the exciting area of electrochemistry, where we develop and use microscopy tools to study the electrochemical interface. Two of the projects in our current research are the study of nanoparticle collision and oxidation behavior, and the nucleation and growth of nanobubbles, all at the electrochemical interface. In a more recent study, we also developed tools where we can detect coronavirus particles one at a time.
I'm at a modified dark field microscope where we use light scattering to look at electrochemical processes. Specifically, we're interested in imaging nanobubble formation on a particle going through a nanopore.
Here is the setup for total internal refraction microscopy. We utilize the evanescent wave to image surface nanobubbles from water reduction by the transient absorption of fluorophores at the bubble surface.
This is the part of the lab where we do feed imaging, which is used to spatially map microscale redox landscapes using electrochemistry. My major role in the project has been to develop a way to improve the sensitivity and spatial resolution of the technique using strictly electrochemical methods.
I primarily work with these carbon bipolar ultramicroelectrode arrays that I fabricate in a clean room here on campus. These are made up of about 150,000 different 8-micron carbon electrodes that are embedded in a polymer membrane. I'm using these right now with this setup over here to electrodeposit a gradient of catalytic metals across the surface. So what this allows us to do is very, very highly parallelized screening of the electrocatalyst that is deposited on each individual electrode.
I'm interested in leveraging the electroactivity of metal nanoparticles to answer interesting biological questions, such as the size of pore openings during exocytosis and neurotransmission. In addition to this work, I also work at the Washington Nanofabrication Facility, making nanoscale arrays for imaging experiments.
This is the amperometry room, where a lot of fundamental research in our lab gets started. By using those fundamental knowledges, we're able to transition to more applicable projects. For example, we're trying to develop new electrochemical-based biosensors to detect the COVID-19 virus.
We're currently in the fabrication room where much experimentation begins. I selected the University of Washington and Bojang's lab as the group moves beyond traditional electrochemistry. The group specializes in coupling electrochemical measurement with optical microscopy to consider entities as small as single molecules. Above all, the group's inclusivity and camaraderie is unparalleled.